Well, thank you very much, everyone, for joining in this colloquium from the Faculty of Behavioral Movement Sciences. Today we have a speaker from our own faculty, which is Richie Goulding. He joined our group in the MyoLab last October, but he actually already approached me more over a year ago, around the time of the start of the pandemic, when he wanted to work with us in Amsterdam on a project to better understand the limitation of exercise capacity. Richie is a very experienced and well-published exercise physiologist who did his undergraduate and PhD degree in the UK, and then moved over to Kobe in Japan, where he almost crossed his paths with Nicola Garrett. You may still know her from her time at the FU. And now he's here in Amsterdam until at least next year. We got a little bit of money to start a project on type 1 diabetes and exercise capacity and glucose signaling, and he will talk about this as well during this seminar. I already see someone in the chat about an announcement, and I think it's an announcement from Huub for the research master students, so you need to type your name in the chat to him directly. This also is a good way to mention that if you have questions, please use during the chat, during the colloquium, use the chat function. If there are a lot of questions, we can then highlight the most important ones during the discussion at the end, and or not. You can unmute yourself towards the end of the colloquium for a verbal question, so to say. Right, without any further ado, Richie, the whole bandwidth of the internet is all yours, and good luck.
Well, thanks for the introduction, and thanks everyone for coming here to listen to my talk today. So as the title suggests, I'm going to talk to you today about limitations to exercise capacity in health and disease. Just going to give a quick overview of what I'm going to talk to you about today. Rob's already done it, but for those of you who don't know me, I'm just going to give a brief introduction of myself. Then I want to start by defining what is exercise capacity and answering the question, why should we study it? So for those of you not familiar with the field, hopefully I'll be able to convince you that it's something worth studying after this presentation. Following that, I'll give a brief overview of the methodologies that I've used in my career so far. The reason I've highlighted this here is that because I think you can still arrive at sound scientific conclusions that have great relevance for patients and the general public alike with the basic equipment that's available in most exercise physiology laboratories. I'll then summarize some of the findings from my PhD that were conducted in young, healthy individuals. We use these findings as a basis for later investigations in some clinical populations, so I'll discuss that next. And then I'll wrap up with some general conclusions. So yeah, as Rob mentioned, I'm from the UK, small town called Bournemouth on the south coast. From 2011 to 2015, I studied in the University of Exeter and did my PhD in Liverpool from 2016 to 2019. I was lucky enough to win a fellowship towards the end of my PhD to go to Japan, where I stayed for a year. And then I moved here in the start of October, where I've been since, as my second postdoctoral position. So, exercise tolerance. What is it and why should we study it? That's the question I want to answer before proceeding. So exercise tolerance is defined as the ability to produce enough muscle force or power to complete a given task. But when we talk about exercise tolerance in clinical populations, we're generally talking about the ability to complete the tasks involved in everyday life, work, or leisure activities. Unfortunately, in many groups of patients, so people with diabetes, heart failure, COPD, exercise tolerance can become so severely reduced that it reduces the ability to complete everyday tasks. So in the extreme, this can mean not being able to walk up a flight of stairs or go to the shops. And as such, this exercise limitation becomes one of the primary factors that reduces these patients' quality of life. So whenever we stress a system like the human body to its maximum, we can learn a lot about its integrated physiological function, both in health and disease. And that's one of the reasons why I find this topic so interesting, because exercise tolerance underpins both elite endurance performance at one extreme at the other. So this study represents for me probably the best piece of evidence that I know that shows that exercise tolerance is something that we should endeavor to study more. It's conducted by Myers et al., published in the New England Journal of Medicine. And these authors conducted a longitudinal study of over 6,000 people. And they followed them up after 14 years. And they showed that survival after 14 years was greatest in those with the highest exercise capacity. So if you look at the figure on the bottom left of your screen, on the y-axis, we have the percentage of the original cohort surviving. And on the x-axis, the number of years of follow-up. And you can see that those with the highest exercise capacity survived at a much higher rate when compared to those with lower exercise capacity. And this relationship also persisted within subjects that had cardiovascular disease that was ongoing. So subjects with the highest exercise capacity with cardiovascular disease survived at much higher rates than those with lower exercise capacity. They also found that exercise capacity had additional predictive power over and above traditional risk factors for cardiovascular disease. So you can see this in the figure here. So we've got history of hypertension, COPD, diabetes, smoking on the x-axis, and the relative risk of death on the y-axis. And the subjects are grouped again according to their exercise capacity, with the highest in the dark gray and the lowest in the light gray. And you can see that exercise capacity has superior predictive power over who survives when compared to all of these other risk factors. So these authors concluded that exercise capacity is a more powerful predictor of other established risk factors for cardiovascular disease in men. And these findings alone show me that resolving the physiological determinants of exercise tolerance is clearly a scientifically important task. So on the next couple of slides, I'll try and explain to you how we do this. So this diagram, I'm sure if you have an exercise physiology background, many of you have seen before. And this is the late Carl Wasserman's eponymous Wasserman's gears diagram. I think it's one of the best images displaying how the body works during exercise, and it still influences how we interpret data in exercise physiology today. So it picts the body as a series of cogs or gears, and it shows the path of oxygen down through the lungs, through the cardiovascular system, and down into the mitochondria where it's ultimately consumed. Conversely, we have carbon dioxide production and its path back to the lungs where it's expired. The thing I like about this diagram is the fact that it makes an analogy between the body and a working mechanical system. So we want to define which parts of the body or which systems constrain our ability to perform exercise in various populations. So how do we do that? Well, we treat the body as a system made up of various mechanical parts, and then we stress the system, or we spin the gears in various ways. And once the gears are spinning, we can measure the physiological responses to exercise. And based upon the responses that we see, we can determine which of these gears are spinning the slowest, or rather which systems are slowing the overall rate of the response. Broadly speaking, this is still how we define limitations to exercise capacity in exercise physiology today, almost 60 years after the introduction of this diagram. One really important variable that helps us assess how each of these gears are spinning is oxygen uptake kinetics, or VO2 kinetics. And that's what I'll describe on this slide, because that's something I've used a lot in my own research. So at the onset of exercise, the ATP demands in the working muscles increase pretty much instantly. So that's depicted by the dashed line here. So we transition to a new exercise intensity, and the demand in the muscle increases instantly. But if we measure oxygen consumption, either at the mouth or at the muscle, the rate doesn't increase instantly, but does so with finite exponential kinetics. So there's a big delay. So during this transition phase, when VO2 is rising to meet the steady state requirements, Any energy requirements that are not met from oxygen consumption must be met via non-oxidative sources. So we term these sources the oxygen deficit. And the oxygen deficit is depicted by the area in between this dashed line and these curves here. The three colored curves represent the oxygen uptake responses for exercise in three different individuals. So one with a very fast response, that's shown by the blue lines, one with an intermediate response, and the red response showing an extremely slow response. And we can see that the speed of the oxygen deficit, or the speed of the VO2 kinetics, has a large impact on the size of the oxygen deficit. And that's really important for the ability to tolerate subsequent exercise, because a larger oxygen deficit will lead to a greater buildup of metabolites related to fatigue within the muscle and predispose towards poorer exercise capacity. Just so you understand for later in the presentation, we characterize each of these oxygen uptake curves by fitting them with an exponential equation, which is shown here. And the equation is defined by a time constant, which essentially shows how fast the rate of increase in oxygen uptake is. So a small time constant reflects a very fast response, and a large time constant represents a very slow response. And importantly, it's been shown that these oxygen uptake responses and curves, when measured at the mouth, accurately reflect the equivalent responses in the muscle. So determination of VO2 kinetics provides us with a window into metabolic control. And when we use this in conjunction with other measurements, it can really help define exercise limitation in various populations. Another tool that I've used quite a lot in my career to aid interpretation of these VO2 responses is near-infrared spectroscopy, or NIRS. So basically, we attach a small probe to the subject's skin just above the exercising muscle. The probe contains a light source and a light detector and radiates near-infrared light into the tissue. So importantly for this technology, hemoglobin preferentially absorbs different wavelengths of light, depending on whether or not it's bound to oxygen. So if you look at the figure on the right here, if we radiate light of a wavelength of 750 nanometers into the tissue, most of it's going to be absorbed by deoxygenated hemoglobin. Whereas when the light source is above 800, most of it's going to be absorbed by hemoglobin that's carrying oxygen. So the light propagates through the tissues, and depending on the concentrations of each form of hemoglobin, this will reduce the light intensity. And based upon how this reduction in light intensity differs as a function of wavelength, we can then determine the concentrations of oxygenated and deoxygenated hemoglobin within the tissues. The reason NIRS is so useful to exercise physiologists is because the signal coming from the deoxygenated form of hemoglobin basically represents the amount of oxygen extraction coming from the muscle. So in the image on the left of your screen, I've got the Fick principle, which dictates that muscle oxygen consumption is a function of blood flow, represented by Q, and oxygen extraction, which is represented by this term here. When we rearrange the equation, however, muscle oxygen extraction is determined by the ratio between muscle oxygen consumption and muscle blood flow. And this is really useful because if the NIRS signal represents oxygen extraction, we can use that to interpret what the causes for any changes have been following an intervention. So for instance, if muscle oxygen consumption is enhanced to a greater extent than blood flow, then the oxygen extraction from the NIRS signal will be greater because it's essentially a ratio. Alternatively, if muscle blood flow is improved to a relatively greater extent than oxygen consumption, then the oxygen extraction signal from NIRS will be lower. So we can see this illustrated in the figure on the right here, which is from some of my colleagues in Japan. So we've got the NIRS responses in one individual in different muscles to the same exercise bout. We've got the responses in superficial muscle, so close to the surface, shown in the white, and the responses in deeper muscle regions, shown in triangles. So deeper muscle tends to have greater rates of blood flow because it's closer to major arteries, and it also has a much greater amount of capillaries when compared to the superficial muscle. So as you would expect from this, the deoxyhemoglobin response to this exercise challenge is markedly attenuated in the deeper compared to the more superficial regions. And we also have a slight slowing of the response. And this reflects the better oxygen delivery in the deeper muscle regions. So with appropriate data interpretation, NIRS and VO2 kinetics can be used in conjunction to provide answers to questions of exercise limitation. So now I want to show you how I've used some of these techniques to answer a physiological question. So one thing that's attracted much attention in exercise physiology is the priming effect. So this effect shows that when you conduct two identical bouts of high intensity exercise, the oxygen uptake response is faster in the second compared to the first bout. So in the figure here, we've got two bouts of 300 watts exercise separated by 6 minutes of recovery. We've also measured the oxygen uptake responses. And then when we superimpose them or lay them on top of each other, we can see that the response in the second bout is much faster. And this is fascinated many in the field because the factors that are responsible for the speeding of VO2 kinetics in the second bout must also be responsible for the control of muscle oxygen consumption. But what these factors are has remained a matter of intense debate. So one clue is that the priming effect appears to be more apparent in situations where oxygen delivery to the working muscle is impaired. So this suggests that the speeding of VO2 kinetics in the second bout might be related to improvements in oxygen delivery. We wanted to test this idea in one of my studies in my first postdoc in Japan. So what we did was conduct a comparison between upright and supine exercise. So when you exercise in the upright position, the working leg muscles are assisted by the effect of gravity. So there's a column of blood that sits on top of the muscle, which provides some added pressure, which aids diffusion of oxygen into the muscle. So when young, healthy individuals do, or young, at least very fit individuals do priming exercise during upright cycling, the priming effect is typically quite small or even non-existent at times. So in the right-hand figure here, the black circles are the VO2 responses in bout 1, and the white circles are the responses in bout 2. And this is in the upright position. And you can see there's only a tiny change in the speed of the response. On the other hand, if you exercise whilst lying down in the supine position, there's no column of blood assisting oxygen transport. And as such, VO2 kinetics becomes slowed, and the magnitude of any priming effect is much larger. So we've got the same in this figure here, and you can see in bout 2 compared to bout 1, the speeding is much more substantial. And also, we found that in bout 2, there's no difference between the upright and supine positions. So this seems like pretty good evidence for improvement of an oxygen delivery limitation. So we had oxygen delivery was poor to begin with in the supine position. We did priming. VO2 kinetics sped up to the same as what they are in the upright position, suggesting that the priming corrected for the oxygen delivery limitation. But did it really correct for the lack of gravity or the lack of the gravitational effect in the supine position? We weren't so convinced that it could do so because we think the magnitude of improvement in oxygen delivery is probably not big enough. So we used a high-powered time-resolved NIRS system to see what was going on with muscle oxygen extraction. So we reasoned, if you think back to the Fick principle on the other slides, if the improvements were primarily due to oxygen delivery, we'd expect a greater increase in muscle blood flow compared to oxygen consumption, and hence oxygen extraction should decrease. Alternatively, if the priming effect's due to intracellular factors, we'd expect a bigger increase in intracellular oxygen utilization relative to the change in blood flow, and hence oxygen extraction should increase. So the latter is actually exactly what we found. So what we have here is the muscle deoxyhemoglobin responses to 2 bouts of exercise. The dashed lines indicate the onset and offset of the first bout, and the onset and offset of the second bout of exercise. White circles are the supine position, black circles are the upright position. And we also have the responses in 3 different muscle groups. So we've got a deep region of the vastus lateralis, a superficial region of the vastus lateralis, and a superficial region of the rectus femoris. You can see in the first bout, consistent with what you'd expect in a situation where oxygen delivery is impaired, the oxygen extraction response is much higher in the supine position compared to the upright position. However, in the second bout of exercise, oxygen extraction either appears to be a little bit higher or completely unchanged. So this suggests that an improvement in muscle oxygen delivery can't account fully for the priming effect. So here we used NIRS and VO2 kinetics in combination to answer a question that has puzzled quite a few people in my field for a long time. Is the priming effect due to oxygen delivery or something intracellular? And I think these data indicate that it's more related to something intracellular. So now I'm going to segue onto my PhD studies. The first portion of my PhD tried to explain an aspect of exercise tolerance in healthy people. And I looked at a parameter known as critical power. So for any of the next slides to make sense to you, I'll try and explain what critical power is on this slide. So if we got a subject to come to the lab on 4 separate occasions, and we asked them to exercise until they failed at 4 different power outputs, and then we recorded how long it took them to reach task failure and plotted the power output at which exercise was conducted versus the time they could sustain it for, the relationship would look like this. So it would be curvilinear, and the ability to sustain exercise would fall away more sharply at higher power outputs compared to lower power outputs. We then fit a non-linear model to this data. It's a hyperbolic equation, and it has an asymptote. So this curve basically tends towards infinity, and this parameter is known as critical power. Now, this is not just a mathematical parameter, but it also has physiological and functional significance. So when you exercise below critical power, you can sustain exercise for a much longer duration than when you perform exercise slightly above critical power. This is also reflected by the physiological responses you see at these work rates. So in the top right-hand figure, the black circles illustrate a participant exercising about 15 watts below their critical power. And you can see oxygen uptake rises and eventually levels off and reaches a steady state. And hence, this participant could continue for quite a long time. On the other hand, in the white circles, this is about 15 watts above critical power. Oxygen uptake doesn't ever reach a steady state, but rises with time and eventually reaches VO2 max. And at this point, the participant can continue no longer. So we're not able to attain a physiological steady state above critical power. And these responses are also reflected by intramuscular metabolism. So in the bottom figure, we have muscle phosphocreatine content. During exercise just below critical power, again, it levels off and exercise is sustainable. Just above critical power, this was actually only 2 watts above critical power. We have an unsustainable response that decreases with time and reaches a minimum value just before exhaustion. So the question I wanted to answer in my PhD is, why can we not attain a steady state above critical power when we can below? What's so special about critical power? So we started off by asking, maybe it's related to the VO2 kinetics during exercise. So during exercise performed below critical power, where we eventually attain a steady state, the oxygen deficit can also stabilize because the energy demands of the task can be completely met via oxygen consumption. During exercise above critical power, however, VO2 rises continually with time, so a steady state can't be attained. Because the oxygen consumption response is always lagging behind the energy demands of the muscle, this means that the energy demands of the task are increasing, and as such, the oxygen deficit is continually increasing during the exercise as well. As the speed of the VO2 kinetics determines the size of the oxygen deficit, what if this effect is related to the attainment of some critical threshold of oxygen deficit accumulation during exercise? Once this threshold is exceeded, homeostasis can no longer be maintained, which would drive inexorable changes in muscle metabolism and oxygen consumption. And this is the hypothesis that I tested during my PhD. So I'm just bringing you back to this figure that I showed you on one of my early slides here, and I'll just revisit the oxygen deficit because it's quite critical to understanding these concepts. So we transition to a new metabolic rate, and during the transition phase, the difference or any energy demands not met by oxygen consumption have to be met by non-oxidative sources. The figure on the right shows you the relationship between the size of the oxygen deficit and the size of the increase in metabolic rate, or we can call that exercise intensity. So a bigger increase in exercise intensity means a bigger oxygen deficit. But you can see that the slope of the line is also a function of the speed of the VO2 kinetics. So a faster VO2 increase means that the slope of the line is lower. So for a given increase in metabolic rate, the oxygen deficit is much smaller. For an individual with very slow VO2 kinetics, a very small 1 liter increase in metabolic rate leads to a much larger oxygen deficit accumulation. So we said, what if there's some critical threshold above which the accumulation of the oxygen deficit doesn't stabilize and its accumulation accelerates? That would then explain an inability to maintain homeostasis above critical power during exercise. So this hypothesis would predict that if you have a faster VO2 kinetics, then critical power should be higher. So does the correlational data support such a link? The first study to look at this was from Harry Rossiter's group in Leeds. They determined the speed of the VO2 kinetics and critical power in a group of 14 individuals and showed a really strong inverse relationship. So those with the fastest VO2 kinetics had the highest critical power, and those with the slowest VO2 kinetics had the lowest critical power. The lead author of this group then conducted a review paper where he expanded this relationship out across the extremes of aerobic function. So in here we've got trained athletes, active, healthy young individuals, healthy elderly people, and COPD patients. And you can see that even when extended across the extremes of aerobic function, this relationship is still strong, inverse, and linear. Obviously, correlation doesn't mean causation, but other parameters of aerobic function typically have much weaker relationships between themselves. So this was quite promising. But to see if this relationship is causal, we needed intervention studies to demonstrate a potential link between these variables. And this is what I did in my PhD. So over the course of 7 studies, we variously used interventions to speed or slow the VO2 kinetics and observe the outcome on critical power. The reason this required 7 studies is because to prove this link, well, the interventions that we used often impacted oxygen delivery, and that's also known to affect critical power. So if we wanted to tease out an independent effect of VO2 kinetics, we had to speed up and slow down the VO2 kinetics, both with and without alterations in oxygen delivery. And that proved to be quite hard to do. But the results across the 7 studies were pretty unanimous. So when we speeded VO2 kinetics, critical power increased. And when we slowed the VO2 kinetics, critical power decreased. These figures are not actually from my work, but I think they illustrate the relationship really nicely. So here you can see 2 individuals with very different VO2 kinetic responses to the same exercise bout. They both have very different critical powers, which are reflected by the intercepts of these linear relationships on the right. We then wanted to, well, given these relationships that we found in health, we wanted to see if this also occurred in a patient population. The one we chose was type 1 diabetes. And there were, well, it would be nice to say that there was a scientific reason for this, but there wasn't really any reason why we chose people with type 1 diabetes at the time. It was just the links we had available with clinicians, and we chose to study them. They don't have any particular exercise tolerance problems, But what we wanted to see is if this relationship in health could be used to enhance exercise tolerance in a patient population, and also tell us something about limitations to exercise tolerance in a diseased population. So we recruited a group of individuals with type 1 diabetes, and we measured their critical power and VO2 kinetics, both with and without a prior bout of priming exercise. So we've got these figures here, which show individuals with type 1 diabetes performing exercise at the same work rate with and without a prior bout of priming exercise. So the white circles are the primed condition and the black circles are the non-primed condition. You can see the VO2 kinetic response was faster following priming, and that's consistent with what we found in healthy people. And here we've got those same VO2 responses, but the final data points show the duration for which exercise could be maintained for. So in the condition where VO2 kinetics were faster, when we did priming, exercise could be sustained for about 200 seconds longer when compared to the non-primed condition. We then looked at critical power. And in this figure, we've got the solid black bars illustrate the group means of critical power between conditions. The dashed black lines are the changes of each individual participant. And we found that following priming, critical power was greater in every patient that we looked at. So this confirmed our findings in health and raised the possibility for interventions aimed at enhancing VO2 kinetics for improving exercise tolerance in patient populations. We then looked at the muscle deoxygenation responses assessed via NIRS to see which systems appear to have been most improved following the exercise bout. So muscle deoxygenation responses to moderate exercise in healthy controls. So this was a matched control group in the black circles and diabetic patients in the white circles. You can see that the deoxygenation response is actually slower in the patients with type 1 diabetes. And this is quite surprising for us because a slower muscle deoxygenation response to an exercise challenge either indicates that muscle oxygen delivery was relatively enhanced in the patients, which seems pretty unlikely, or there was a relative deficit in intracellular muscle oxygen utilization. And the findings from priming actually confirmed this. So in the right-hand figure, this is just the type 1 diabetes group now. So the white circles are the same responses that you see in the diabetics in the left-hand figure. And the black circles are the responses following priming. And we saw a speeding of the muscle deoxygenation kinetics following prior exercise. So this suggests that oxygen utilization was enhanced to a relatively greater extent than oxygen delivery, suggesting that in contrast to many other clinical conditions, the limiting factors for exercise tolerance and VO2 kinetics in type 1 diabetes reside intracellularly, as opposed to being related to oxygen delivery. So our non-invasive findings hinted towards impaired mitochondrial function being the primary constraint to exercise capacity in individuals with type 1 diabetes. Pardon me. This suggestion was then subsequently confirmed by studies using muscle biopsies that directly assessed mitochondrial function in individuals with type 1 diabetes. So you can see this illustrated in the two figures at the top here. So the red staining indicates the mitochondria, and we have the control subjects on the left and an individual with type 1 diabetes on the right. You can probably make out that the individual with type 1 diabetes has very disorganized mitochondria, and the crista are really fragmented and disorganized. So in the lower left-hand figure, we've got the rates of mitochondrial respiration when various concentrations of ADP are applied. So ADP is an extremely important substrate for mitochondrial respiration. This will tell us something about the function of the mitochondria in these individuals. So this disorganized structure directly translated into lower ADP-stimulated mitochondrial respiration. So these mitochondria were less able to produce energy when compared to healthy individuals. Interestingly, these mitochondrial alterations occurred in individuals with diabetes that were already meeting or exceeding recommended physical activity guidelines. So this suggests that somehow the muscles of individuals with type 1 diabetes are somehow resistant to exercise training. Skeletal muscle mitochondria are involved in many pathophysiological states like insulin resistance, type 2 diabetes, aging, and cachexia. So this means that we need to resolve the causes of this dysfunction because it's important for these individuals. And this is partly what I'm trying to do in my research here at the moment in the VU. Another big problem for individuals with type 1 diabetes is that even if they don't consume insulin before exercise, if they do a prolonged exercise bout, as you can see here in this figure on the left, we've got blood glucose concentrations over time in a healthy individual in the white diamonds and a individual with type 1 diabetes in the gray diamonds. In spite of no exogenous insulin, blood glucose declines with time and will eventually lead to these individuals becoming hypoglycemic. The key question is, how does this occur? And it's really important because exercise is a cornerstone of diabetes management. So it's really important for these individuals to exercise because it really improves their condition, but it comes with the barrier of actually leading to a hypoglycemic event. So how can we figure out how this works and how we can prevent this? This is shown in the figure on the right here. And you can see that muscle glucose uptake is led by a transporter known as GLUT4. There are many different pathways that lead to the translocation of GLUT4 to muscle membranes, causing glucose uptake. There's lots of different pathways and they're not very well defined. And because muscle glucose metabolism or whole body glucose metabolism is altered in type 1 diabetes, it stands to reason that these pathways will also be altered in diabetes. So what I'm trying to do in my current project also is investigate which of these pathways are predominant in skeletal muscle in type 1 diabetes. And then we can get a better understanding of how to prevent hypoglycemic episodes during exercise in these individuals. So yeah, this is my current project. We're assessing two things, as I said, mitochondrial function and muscle glucose transport in individuals with type 1 diabetes before and after exercise training. So during visit 1, subjects come to our lab for a ramp exercise test on a cycloergometer. We use some of the measures that I've showed in previous slides, like NIRS, and this will help us to determine the limitations to their exercise capacity. Visit 2 then takes place at the hospital, and this involves a 30-minute bout of moderate intensity cycling exercise. Before and after the exercise, we take blood samples and muscle biopsies. The purpose of the exercise bout and the biopsies before and after is so that we can stimulate muscle glucose uptake with the exercise and see exactly how it's being transported inside of the muscles during exercise. Following visit 2, we then have a 4-week training intervention. So that's 3 times per week of moderate intensity cycling exercise, and it goes from 30 minutes in week 1 per session to 60 minutes in week 4 per session. And then after the training, subjects return to the lab for visits 3 and 4, which are exactly the same as visits 1 and 2. We want to see if exercise training will improve their mitochondrial function and how any improvement might occur. Studying this will hopefully give us some insight into why their mitochondria are impaired in the first place. Oh, too fast there. Yeah, so this study is ongoing at the moment. And as you can see from the picture, I've kindly volunteered to run through the exercise protocol as myself as the first subject to check that everything is running smoothly and safe for our patients. So fortunately, my muscle biopsy went fine, and we're now actively recruiting for patients. So please, if anyone knows any friends or family members with type 1 diabetes who'd be interested in taking part, then please reach out to me after the presentation. I'll be happy to have a chat. Okay, so if I have time, another project I've been working on since I've been at the VU is on limitations to exercise capacity in patients with multiple sclerosis or MS. So on the last few slides, I'll just give a brief overview of the findings of this project. MS is a disorder of the central nervous system, where the immune system attacks the myelin sheath that surrounds nerve fibers, and this causes communication problems with the rest of the body. So the symptoms are really varied, but they can include vision problems, cognitive deficits, coordination problems, and a reduction in overall physical function, which is what I was obviously most interested in. The reduction in physical function can also limit the activities of daily living, which predisposes these people towards a sedentary lifestyle and puts them at a greater risk of various lifestyle-related diseases such as cancer, cardiovascular, and respiratory diseases. The causes of this reduction in exercise capacity are unclear, and the observations are relatively scattered, and there's no conclusive evidence regarding what defines exercise limitation in these patients. And another tricky thing to get around is the fact that these patients typically exercise less. So it's really difficult to tease out whether or not the exercise limitation is because they exercise less or because it's a primary effect of the disease. So in this study, we matched about 20 MS patients with age and sex and activity matched controls. And we got them to perform incremental ramp exercise, so non-invasive exercise testing. And we also subjected them to muscle biopsies to see what was going on inside the muscles. So the bit that we can't access with our non-invasive measurements. If you remember back to one of my early slides, where I showed a picture of Carmen Wasserman, he also designed this famous figure called the 9-panel Wasserman plot. Basically what it is we measure a number of physiological variables during incremental ramp exercise. Some of the panels correspond to pulmonary function, some correspond to cardiac function, some correspond to metabolic function, and there's still more others. But this is a really famous figure that enables systematic interpretation of responses to exercise. And this is the approach that we employed in this study. So we compared the MS patients with the controls from their performance on an incremental ramp exercise test. And we found that, as we expected, the MS patients had a reduced maximal exercise capacity. So VO2 max was reduced, and also many submaximal parameters of aerobic function were also impaired. So no surprises, but that was consistent with the previous literature. What this 9-panel plot enabled us to do, and I don't really have time to go into it into detail, but it revealed to us that there was no clear impairment in cardiovascular or pulmonary system parameters. So we couldn't explain all of the exercise impairment via anything to do with the cardiovascular or pulmonary system. So this meant to us that the skeletal muscle impairment or the skeletal muscle must have been contributory to the exercise impairment. So to assess the possibility of a muscle metabolic contribution to the exercise impairment in these patients, we next assessed skeletal muscle fiber type composition and size. The stainings are illustrated at the top. So we've got type 1 fibers shown in the blue, 2A in the green, and 2X in the red. And as you can see from the bottom left figure, there were no differences in the total percentages of fiber type composition between patients in the blue and controls in the white. The significance symbols you can see here are differences between type 1 and type 2 percentages, not between the patient and control groups. In the bottom right, we can see that there were no differences in fiber cross-sectional area for any of the principal fiber types assessed between the patients and controls. So then we concluded that the exercise limitation in these patients wasn't due to differences in fiber type, composition, or fiber size. Next, we assessed muscle fiber oxidative capacity. So we inferred this by succinate dehydrogenase activity, or SDH, between the groups. So in the left-hand figure, you can see a reduction in fiber SDH activity across all 3 fiber types in the patients versus the controls. The middle figure shows the reduction in SDH activity weighted across all the fiber types. And you can see that the effect still persists. And in addition to oxidative capacity, we also found that the capillary density of the MS patients was reduced when compared to controls. So we concluded that a reduced mitochondrial oxidative capacity or peripheral oxygen supply could potentially contribute to exercise limitation in these patients. So to provide further insight into which variables were the primary determinants of reduced exercise capacity in MS, we next did some correlational analyses. So just to provide some context for these findings, if you correlate VO2 max with muscle oxidative capacity or SDH activity across the extremes of human populations, so heart failure patients at one end and elite athletes at the other, you'll get a really nice, strong linear relationship because you have a wide range in VO2 max and you also have a wide range in oxidative capacity. Whenever you assess these relationships in individual populations, the relationships are normally weak or non-existent. And that's because VO2 max is typically considered to be limited by factors related to convective and diffusive oxygen delivery in healthy individuals. So consistent with that notion, you can see in the left-hand figure that in the healthy controls shown by the black circles, there's no relationship between SDH activity and VO2 max. Whereas in the MS patients, the relationship is very strong, it's positive, and it's linear. I don't have time to go into these two parameters, but unlike SDH activity, we found no relationships between exercise-related variables and muscle capillary density or the capillary fiber ratio in MS, but we did in the healthy controls. So this suggests that in health, consistent with previous work, extramuscular factors are probably more relevant for determining VO2 max. Whereas in MS, we think that there's an intramuscular limitation constraining exercise capacity. Okay, final piece of evidence on this slide. My circles appear to have drifted somewhat, which is annoying. But here we have a regression model. So we've done a multiple regression model to try and explain which factors contribute to VO2 max most in healthy controls and MS patients. So we'll start with the model in healthy controls. So you can see the model overall explains about 64% of variance in VO2 max. But the only significant contributor to this model was the capillary to fiber ratio. So peripheral oxygen supply and O2 diffusion appears to be an important factor limiting exercise capacity in health. On the other hand, in MS, capillary fiber ratio didn't contribute to the model, which explained 71% of the variance in VO2 max. The only contributor was the muscle fiber oxidative capacity. So just to firm up on those conclusions, clearly, I think based upon these findings, the skeletal muscle mitochondria appear to limit exercise tolerance in these MS patients. I don't have time to go into it, but we also did an exercise training intervention to see if the impairment in exercise tolerance was related to physical activity or exercise or something primarily related to the disease. And the muscles of the MS patients actually didn't adapt following exercise training. So that suggests to me that there's an intrinsic factor related to this disease that impairs their mitochondrial function. So I think that the findings from this study highlight that a combined approach of invasive, non-invasive measures can help provide greater interpretive power over either alone. So that's my final slide, and I'll just briefly conclude. So in my career so far, I've worked on trying to explain limitations to exercise tolerance in health and disease. It's my opinion that all too often it's considered that scientific insights that allow translation from bench to bedside only come from the world of omics or big data or overly reductionist techniques. Maybe my conclusion is too grand based upon my own work, probably is, but I think what I've tried to show is that you can still make sound scientific insights into various diseases with standard exercise physiology equipment and well-designed hypotheses. I think obviously a range of techniques, both invasive and non-invasive, provides a more complete picture. And some mechanisms can only be revealed via invasive work. And that's why I came here, because I wanted to learn these techniques. But I guess the take-home message is, let's not forget our old tools just yet, because I think they still have a lot to tell us. So this is a list of all the people that were involved in the studies that I presented today. So a big thank you to them, and thank you to everyone for listening to my presentation.
Thank you very much, Richie. I'll do this. I'll do the clapping in behalf of the 57 other people in the Zoom room. There are certainly time for questions, and I think there should be questions as well from the audience. Are there any? Didn't see anyone in the
Hi Rob, hi Richie. I would like to ask something. I'm really excited about the MS data coming from the MS field, of course.
Okay.
So actually, I was wondering, looking at your acknowledgements, I see Jack van Horst being involved, but are there also some clinicians involved in the study?
Yeah, so this actually was a project from somewhere in Belgium, I think. So I wasn't actually involved in the data collection or the analysis. I analyzed the exercise data.
Okay.
Helped us interpret the.
Okay. It's interesting because dysfunctional mitochondria has certainly been one of the issues as being thought of underlying the cause of MS. But then mitochondrial dysfunction in the brain, of course, in the central nervous system. Of neurons or oligodendrocytes, and now your data suggests that also in muscles there might be some dysfunction in mitochondria. Of course, that's immediately the question, like how can a disease of the central nervous system affect mitochondria in the muscles, or shouldn't we think of it anymore as a disease just of the central nervous system?
Yeah, well, I guess so. I mean, the data I guess when I looked at the previous literature before writing this paper, the suggestion was that this mitochondrial dysfunction or exercise limitation is primarily a function of reduced physical activity. But actually, we did an exercise training intervention and nothing happened to their muscle mitochondria. So clearly, there's some kind of intrinsic aspect.
Yeah, that's what you mentioned. So that's interesting, but I try to understand how come. If it's something that's coming down from the central nervous system, or is it, as I said, is it a disease that we shouldn't look at anymore as just a central nervous system disorder, but that also peripheral tissues are affected by whatever the cause is, leading to this mitochondrial dysfunction at a much broader range of tissues than just within the central nervous system itself. One of the main features, which is widely distributed among MS patients, is fatigue. And that's very difficult to counteract also in the patients. So I thought fatigue could be also due to this problem that you actually notice now in the muscle, that people feel fatigued. I mean, it could be one way or the other. It's the chicken and the egg, of course.
Yeah, we don't have any. Oh, sorry.
It's certainly interesting also to pursue this type of research within our MS center, because I know that, of course, I think he's not around, Erwin Verwegen, he's also, and Hanneke Huls, they're also involved in this type of research as exercise in MS patients. And I think your finding is really, really interesting.
Well, thanks. I think we don't have any concrete answers yet, but we did talk about looking at whether any markers of systemic inflammation were altered, didn't we, Rob? So I don't know if we're wondering if that might play a role, and then that would confirm your suggestion that it's not just a disease of the nervous system. It's something far more systemic. I don't know if you have any comments on that, Rob.
Yes, indeed. I think we so together with Bert op de Heinde, he's from the Hasselt University. We did perform this study, and Jan Spaan, the first one on the list here, and they did have some blood value, some blood measurements, but they didn't assess the inflammatory markers before and after training, whether that was an improvement there. I would have guessed that that would be a contributing factor here. The other thing that we looked into a little bit was whether because of course it's a motor neuron disease, it could happen that you get denervation and re-innervation of the skeletal muscle, and that would cause some specific pattern in the sections which wasn't there. So I think there is really something systemic and not related primarily to the motor neuron function.
But it's not motor neuron disease.
No, exactly. So, but we thought if there would be an
if it would be,
we would see that would explain maybe some of the aspects, but that was not the case. So we think it must be something more related to a circulating factor in MS. And
did you have the tissues that you analyzed? Did you also analyze? Could you be able to analyze other things in the muscle, like?
Yeah, I want to talk. Let's talk about it in a separate.
Okay, fine.
Because I think there will be more questions, and we don't have so much time.
Okay, fine. Yeah,
but I think this is a very good point that we should follow up, Annemarie. I'd like to plan a separate meeting about this.
Okay, perfect.
Are there more questions? Yeah, Hein, I see you're raising your hand.
I have a brief question about the priming effect.
Okay.
There are 2 consecutive exercise bouts, and I assume that in the second exercise bout, the body is warmer than in the first exercise bout. So is it higher temperature of the body affecting the oxygen extraction?
Yeah, I think we've discussed this before, and it seems that in isolated cell models, so Rob's actually published a paper on this, you do get a speeding of VO2 kinetics when you elevate temperature. But the studies that have addressed this in humans are far less conclusive. So lots of people have tried to elevate muscle temperature and see if that can account for the priming effect. And it doesn't really do anything priming-like. So yeah, I guess the evidence is inconclusive to say whether or not it's a big contributor.
Thank you.
Cheers.
I can see a nice bachelor or master project come along here. With your bath, Hein, in the temperature room. Are there more comments or questions?
Yes, Rob.
Yeah.
Yeah. Also related to this priming effect, Richie. Very nice work. But could it be also that there is phosphorylation of the myosin light chains and heavy chains, which makes the muscle having a higher activity of oxygen kinetics, and also the supply, because these capillaries, because of the nitric oxide, will probably be more open, and the supply will be higher. I just came in a bit later, so may have missed this point in your explanation, but
yeah, well, I guess everything almost everything I presented today is from non-invasive measures coming from whole body physiology or small isolated sites. So can't really comment on any specific intracellular mechanisms. But that certainly could be a factor that's involved. Me and Rob are actually currently writing a paper about what some of the intracellular mechanisms might be. So yeah, maybe that's something we could discuss in the future. But yeah, the phosphorylation of the myosin chains is not something I've considered. But yeah, I don't know. How would that work in your opinion? How would that accelerate oxygen consumption in subsequent exercise?
You get faster contractions of the myosin heads. So then that could elicit more power, but also quicker turnover, and probably oxygen consumption could increase.
Well, the issue, of course, is, Richard, I think it's a good point. Maybe there is a factor that relates to that. But if that's the case, then the ATP turnover rates would be higher, and also your steady state VO2 would be higher.
Well, you actually do get higher primary amplitude. So the initial response is a little bit higher. Okay. So you do see a bit of that actually, which is interesting, I guess.
But that could relate to that effect.
Well, maybe something to look into.
Yeah, sure. Thanks.
It's a little bit difficult to mechanistically alter this. How would you do that? Do you have any idea if you can mechanistically alter these light chain?
Go back to the in vitro, the single fiber work. Yeah, maybe.
Are there more questions? One last question, quick question, comment. I have a question.
Can I have a quick question? Richie, thanks for your presentation. What do you expect regarding the training study in your diabetic patients? Because, well, you have now the experience with the MS patients, and there maybe are similarities in if they are have a dysfunctional mitochondria. Wouldn't it be better to have these diabetic patients, because you say exercise is important, but that strength exercise may be more important even than endurance exercise? Can you comment on that?
Yeah, well, I think for any population, obviously a combination of both is better. With respect to what we'll see following the exercise training, I'm really not sure, because the individuals that have been shown to have mitochondrial dysfunction, and also the patients from my own work, which had relatively impaired exercise tolerance related compared to controls, they were already physically active and meeting guidelines. So it suggests that they're working at a relative disadvantage. Whether that means their mitochondria are less responsive, I don't know. I guess we'll find that out. But yeah, perhaps it is a situation similar to the MS patients. Hopefully in a few months' time, I'll be able to give you an answer, a better answer.
Yeah, good comment, Jo. It's a fair point. It was not a primarily answer, of course, of the research proposal, the grant that we wrote. It was more related also to glucose handling, and then strength training is not your typical intervention to alter glucose handling. But I think that's a good point to keep in mind. All right, but I think by this we should finish this this seminar. Thank you very much for joining in or tuning in. Normally there was always a hangout session here. I don't know if there's anyone who has set this up for this colloquium for a sort of online drink session. I didn't see anything in the email or in the announcements. I'm just looking around whether that would be the case if there was. If not, I think I'd like to thank you all for joining in. If you have more questions for Richie, I think it's nice to stick around a little bit if you have a personal question or so for him. If not, thank you very much, and I'll see you back at one of the future colloquia. Bye-bye.
Thanks.